Ribonucleic acid. In this chapter, we are going to learn about their structure, differences between DNA and RNA, genetic makeup of RNA, some aspects of messenger RNA or nuclear RNA, mRNA, differences between prokaryotic and eukaryotic mRNA, informosomes, ribosomal RNA, transfer RNA, and functions of different types of RNA. Ribonucleic acid or RNA is single-stranded nucleic acid found in all living cells. Its different forms are associated with the transmission of information from nucleus into the cytoplasm and with the synthesis of proteins for the regulation of cell activities. However, DNA is the only macromolecule which also functions for the storage of information and also as a catalyst. The catalytic RNAs are called ribozymes. RNA is also presumed to have played an important role as an essential chemical intermediate in the evolution of life on this planet. RNA is found chiefly in the cytoplasm but also in the nucleolus. Inside the cytoplasm, it occurs freely as well as in ribosomes. RNA can also be detected from mitochondria, chloroplasts, and associated with the eukaryotic chromosome. In some plant and animal viruses, RNA acts as a hereditary material. Let's learn about its structure. The structure of RNA can be easily understood by comparing it with the structure of DNA. More commonly, RNA is a single-stranded structure consisting of an unbranched polynucleotide gene, but it is often folded back on itself, forming helices. DNA is a double-stranded structure, and its two polynucleotide genes are wound spirally around the main axis. RNA, like DNA, is formed of several hundreds of thousands of nucleotides arranged in linear sequence and connected together by 3' to 5' phosphodiester bonds. The sugar found in nucleotides of RNA is ribose, whereas it is deoxyribose in DNA, and nucleotides of RNA are called ribonucleotides. A molecule of ribose sugar, ribosome, a nucleotide of RNA or uridylic acid, etc. The four nitrogenous bases found in RNA are adenine, cytosine, thymine, and uracil. Adenine, cytosine, guanine, and uracil, whereas those in DNA are adenine, thymine, cytosine, and guanine. Therefore, in RNA, thymine of DNA is substituted by uracil. Some unusual nitrogenous bases also found in RNA. The base composition of RNA does not agree to the AUGC is equal to 1, but intramolecular pairing between the nucleotides of single strand of RNA provides stability to RNA. In DNA, nucleotides of two polynucleotide strands pair through hydrogen bonds. DNA is the hereditary material, whereas RNA are of different types for performing different functions during protein synthesis. DNA is the hereditary material. DNA is a hereditary material, whereas RNA are of different types for performing different functions during protein synthesis. However, in most plant viruses and some animal viruses, RNA also acts as hereditary or genetic material. The genetic RNA. In most plant viruses, some animal viruses, and in some bacteriophages, DNA is not found and RNA is acting as a hereditary material. Thus, RNA may be single-stranded or double-stranded. When it is double-stranded, it follows the same rules of base pairing as in the case of DNA. But when it's single-stranded, it's just remain like that. Then, differences between DNA and RNA. I'm going to read about DNA. Then after that, I'm going to read about ribonucleic acid. Okay. So first, let's start reading about deoxyribonucleic acid. DNA is found in the chromosomes of nucleus and is chiefly concentrated in the nucleus only. DNA is a double-stranded structure having a helical configuration. The two strands are coiled spirally in opposite directions. The sugar molecule in DNA is deoxyribose. The four nitrogenous bases found in DNA are adenine, guanine, cytosine, and thymine. Adenine and guanine, purine bases, cytosine and thymine, pyrimidine bases. The purines and pyrimidines in DNA are found in equal proportion. DNA is the hereditary material, and the information for the proteins needed during different cell activities and life processes are encoded in DNA molecules. Base composition in DNA is AT. A by T is equal to G by C is equal to 1. Next, we are going to read about ribonucleic acid. RNA is chiefly concentrated in the cytoplasm, although it also occurs in the nucleolus and so also in some extent to nucleoplasm, where it is associated with the cytochromosomes. So the RNA molecules are single-stranded. The strand, in some cases in tRNA, is folded on itself, and in the folded region, the nucleotides may be connected by hydrogen bonds. The single sugar molecules in RNA is ribose. The 4 nitrogenous bases found in RNA are adenine, guanine, cytosine, and uracil. Adenine and guanine, purine bases, cytosine and uracil, pyrimidine bases. In RNA, thymine is replaced by uracil. It is not necessary that purines and pyrimidines occur in equal proportions, unlike DNA. RNA participates in protein synthesis. Base composition in DNA is A by T is equal to G by C is equal to 1. We mentioned earlier that base composition is like that 1, but it is not so in RNA. Replication of genetic RNA. Viruses follow reverse transcription. In retrovirus, viruses having RNA as hereditary material possesses an enzyme called reverse transcriptase. When a virus particle enters the host cell, its RNA is released in the host cytoplasm. Here, with the help of reverse transcriptase, a strand of complementary DNA is synthesized using RNA as a template. This single DNA strand synthesis is complementary anti-parallel strand completing the formation of double-stranded DNA. This viral DNA then joins the DNA of host cell, becoming its integral part. The integrated DNA copy of host DNA is called provirus, and this process of DNA transcription is called reverse transcription. Then, utilizing synthetic machinery of host cell, this DNA produces several copies of RNA and protein molecules to be used in the form of viral protein coat. RNA, ssDNA, dsDNA, viral mRNA, viral proteins. Types of viruses: plant viruses, animal viruses, bacteriophages. Names of viruses, for example, TMV, tobacco mosaic virus, then worm tumor, influenza virus, poliomyelitis, rotavirus, MS2, F2, R17, etc. are some sort of viruses which contain RNA as genetic material. So first, replication of viral particles of retrovirus by reverse transcription inside host cell. When we try to learn about this procedure, we could see attachment virus attaches to host cell. Penetration. Next step, penetration. Virus loses envelope and capsid and release RNA. And reverse transcription. Reverse transcription synthesizes a DNA strand on viral RNA. Then replication, synthesis of double-stranded DNA. Then DNA integration. DNA integrates into host chromosome. Transcription. Several RNA molecules transcribed inside host nucleus. Then translation. Transcribed RNA inside host acts as an mRNA for the synthesis of coat protein, envelope protein, and viral RNA. Translation. Then maturation. Assembly of new viruses. Liberation. New viruses liberated by exocytosis. These are the major processes. In RNA viruses, this virus synthesizes new genome RNA on RNA template with the help of an enzyme called replicase. This new RNA molecules then synthesize mRNA, which guides the synthesis of viral proteins using host's ribosome of the RNA, amino acids, and required enzymes. So RNA, mRNA, polypeptide, like that. Earlier we mentioned about the case with retroviruses. It's like RNA transforming to ssDNA, then dsDNA, viral mRNA, and viral protein. Here we are talking about RNA viruses. RNA, mRNA, polypeptide. That is the process. Then non-genetic RNA. We just learned about genetic RNA. Now we are going to learn something about non-genetic RNA. In all other organisms where DNA is the hereditary material, all types of RNA are non-genetic. The non-genetic RNA are synthesized from DNA template. In general, 3 types of RNA have been distinguished: messenger RNA or nuclear RNA, messenger, ribosomal RNA, transfer RNA. When we shortly represent all this, we often represent messenger RNA as mRNA. mRNA in the sense, a very basic idea, like the capital RNA. RNA is written in capital letters and the M will be in small letter. Messenger representation M, small m, capital RNA. Messenger RNA. Then ribosomal RNA, small r RNA. Transfer RNA, small t RNA. Never replace that small t with capital T, you will end up having a mistake. Okay. Then messenger RNA or nuclear RNA, we are going to learn about this. So messenger RNA or nuclear RNA carries genetic information from chromosomal DNA to the cytoplasm, where it acts as a template for protein synthesis. It is complementary to DNA and carries the copy of the same base sequence as found in that part of DNA from which it is copied, except that thymine is substituted by uracil. It constitutes about 5% of total RNA of the cell. Since the genetic blueprint is contained within the nucleus and the work benches or sites of protein synthesis, ribosomes, are present in the cytoplasm, this RNA molecule serves to transport genetic messages from DNA to ribosome. The term messenger RNA was used by Jacob and Monod in 1961, two scientists. In the cytoplasm, mRNA molecules become attached to ribosome and act as template for the polymerization of amino acids into polypeptide chains. The sequence of amino acids in each polypeptide chain is determined by the sequence of codons in mRNA molecule. Characteristics of mRNA. mRNA has following characteristics. This is formed as a complementary strand to one of the two strands of DNA. It therefore carries the same sequence of base arrangement as found in the part of DNA from which it is copied, except the thymine of DNA is substituted by uracil mRNA. mRNA therefore contains the same information as coded in that part of DNA which is acting as a template. Then after synthesis, it immediately diffuses out of the nucleus into the cytoplasm, where it gets associated with the ribosome. Here mRNA acts as a template for protein synthesis. Then mRNA has a short lifespan and withers away after few translations. It is a high turnover. mRNA from different organisms may be alike in gross base content, but they usually differ in the arrangement of bases. The molecules of mRNA are linear and longest of all the 3 types of RNA. Their length is related to the size of polypeptide chains for which these have coded information. There is one mRNA for each polypeptide chain. Because of great variation in the size of mRNA molecule, mRNA is also called heterogeneous nuclear RNA or hnRNA. Heterogeneous nuclear RNA. Then structure of mRNA. The main part of mRNA is the sequence of nucleotides that code for a polypeptide. However, a fully processed mRNA molecule has at either end some sequences that are not translated. In case of these sequences, 2 ends of each mRNA molecule are distinctly marked. Each fully processed mRNA molecule has following parts: a cap of methylated guanine at its proximal or 5' end, G cap at 5' end, a start or initiation codon AUG or GUG at this G cap proximity. So leader sequence, ribosome binding site, start codon AUG, coding sequence, Taylor or non-coding segment, stop codon. These are the portions of a prokaryotic mRNA. But when I try to learn about eukaryotic mRNA, you could see some differences. Methylated G is there, leader sequence, ribosome binding site, initiator codon AUG, coding sequence, termination codon, then untranslated region will be there, and a poly A tail at the 3' end. These are very important portions, and a fully processed mRNA with marked ends. We just this structure. Then some other aspects related to mRNA that contain a long coding region, a termination codon or stop codon, UAG or UGA, at the end of coding region at the distal end, 3' end most probably. and a poly A tail of many adenine nucleotides. A small non-translated or untranslated segment, one may be present after the G cap and before poly A tail. The non-translated sequence at 5' of mRNA is called the leader because it precedes the start of initiation codon. This leader segment has ribosome binding site where mRNA binds to ribosome. The non-translated sequence at 3' end is a Taylor or non-coding segment 2. It follows the termination codon. Though leader and Taylor sequence are not translated, their presence is essential for the translation of message coded in the coding region. Difference between prokaryotic and eukaryotic mRNA. Character nature. Prokaryotic mRNA may be polycystonic or monocystonic, but eukaryotic mRNA is always monocystonic. Then 5' methylated G cap, most probably absent in prokaryotic mRNA but present in eukaryotic mRNA. Ribosome binding site or Shine-Delgado sequence present in prokaryotic mRNA, absent in eukaryotic mRNA. Shine-Delgado sequence. If you learned about the transcription and translation processes of DNA or replication process, you might have learned about this Shine-Delgado sequence. If not, just try to learn this topic by just stretching. Then poly A tail at 3' end, prokaryotic mRNA absent, eukaryotic mRNA present. Poly A tail at 3' end. And heterogeneity of mRNA. The molecules of mRNA are heterogeneous because this occur in different sites having different molecular weight. The heterogeneity depends upon two main factors, the size and number of cistrons and the size of protein molecule for which it carries codons. Based on the number of cistrons, two types of mRNA have been recognized. Monocistronic mRNA molecule containing the codons of a single cistron, which code for one complete molecule of protein. In eukaryotes, mRNA carries information only for the polypeptide gene. This monocistronic or monogenic because it is transcribed from a single cistron gene and has a single initiation codon and a single termination codon. But polycystonic mRNA is also there. Molecules contain the codon for more than one cistron, which may lie close together. This type of mRNA synthesis more than one polypeptide gene. Therefore, a polycystonic mRNA molecule must possess a series of start and stop codons. If a single mRNA molecule encodes 3 proteins, the initial requirement would be the sequence. So start codon, protein 1, stop codon, start codon, protein 2, stop codon. Then again start codon, protein 3, stop codon. These are the three sequences. First sequence, second sequence and third sequence. In such mRNA molecule, the leader sequence preceding the first start signal may be several hundred bases long, and there is usually a sequence called a space region of 5 to 20 bases between one stop codon and the next start codon. The structure of tricistronic mRNA is very important, and we are going to learn that. The mRNA molecule for the metabolism of histidine codes for synthesis of 10 specific enzymes. Let's learn the arrangement of control elements and coding sequence of cistrons in a typical polycystonic lac operon. Promoter region, operator region, start point of transcription, coding sequence, beta galactosidase. For example, here we are going to learn about the lac operon structure. And beta galactosidase coding sequence, then galactoside permease coding sequence 2, coding sequence 3 will be there, that will code for triogalactoside transacetylase, then transcription termination site. Next, biosynthesis of mRNA. Synthesis of mRNA is accompanied by using one of the two DNA strands. It is carried out from the 5' end towards 3' end on 3' to 5' polynucleotide strand. RNA polymerase attaches to the initiation site or promoter end and the structure gene of DNA cistron and catalyzes DNA synthesis. The phenomenon of synthesis of mRNA from DNA is known as transcription. Template DNA, exon 1, exon 2, then intron, Formation of mRNA from primary transcript in eukaryotic cells, template DNA, exon 1, exon 2, exon 3 portions. Intron is also there. Transcription by RNA polymerase 2 and capping primase transcript, 5' end is there. Removal of 3' end by nuclease. Then 15S processing intermediate, polyadenylation will be taking place. After that, endonucleolytic cleavage at splice junctions, then ligation of exons. We know that introns are also there. So we need to remove those non-coding portions, and this just by sealing those exon portions or ligation. There might be some ligases acting on this ligation process. So ligation of exons will be taking place. That will create a 10S mature mRNA. Then in eukaryotes, mRNA is synthesized as heterogeneous nuclear RNA, hnRNA. Here when we write this, hnRNA, heterogeneous RNA, nuclear RNA, that H and N will be represented in small letters, and then RNA as usual capital letters. This is a very basic information. If you don't know, then just try to follow this carefully. Inside the nucleus, these portions are the heterogeneous nuclear RNA. These molecules are much bigger in size than the functional mRNA molecules because these are copies of both introns and exons. They are called primary transcripts. A sequence of about 210 nucleotides of polyadenylic acid, poly A, is added to the 3' end of each primary transcript, and intron segments are cleaved. The transcripts of exon then join. Finally, the 5' end of hnRNA starts disintegrating to release poly A mRNA. These poly A mRNA molecules diffuse out in the cytoplasm. Lifespan of mRNA. Lifespan of mRNA in prokaryotes is not that much long, it's very short. In bacteria, it is about 2 minutes. But in eukaryotes, mRNA is metabolically more active and can function for a number of hours and even for days. Informosomes. The eukaryotic mRNA is associated with the proteins forming ribonucleoprotein complexes. Some of these complexes remain free in the cytoplasm without being attached to the polyribosomes. These were named informosomes by Spirin. They are very stable and can remain in the cell cytoplasm for several days. Their stability is attributed to the presence of protein sheath around each mRNA molecule. In informosomes, proteins and mRNA are found in the ratio 4:1. These occur in the form of particles of 80S or less. Then role of messenger RNA or msRNA. Genetic information for protein synthesis is encoded in the DNA of a gene. It is stored in the nucleotide sequence in DNA present in the nucleus. Protein synthesis occurs in the cytoplasm. The sequence of nucleotides in DNA determines the sequence of amino acids in the polypeptide gene. Messenger RNA, mRNA acts as an intermediate in the flow of information from DNA to protein. mRNA is assembled as a complementary copy of 3' to 5' DNA strand of a gene. Once again, genetic information for protein synthesis is encoded in the DNA of a gene. We know that. It is stored in the nucleotide sequences in DNA present in the nucleus. Protein synthesis occurs in the cytoplasm. The sequence of nucleotides in DNA determines the sequence of amino acids in the polypeptide gene. Messenger RNA or mRNA acts as an intermediate in the flow of information from DNA to protein. mRNA is assembled as a complementary copy of 3' to 5' DNA strand of a gene. It is synthesized in 5' to 3' direction. The 5' to 3' strand of DNA that is not transcribed is called coding strand or anti-template strand. It has the same sequence of nucleotides as the RNA transcript, except that its T is replaced by U in the RNA transcript. This process of formation of RNA from non-coding or template strand of DNA is called transcription. The sequence of nucleotides in mRNA is complementary to that of gene in which it is transcribed. It means that mRNA retains the same information as the gene itself, and the genetic information remains stored in the nucleus and as a part of immobile DNA molecule. It is imparted to a comparatively small and mobile mRNA. This mRNA passes out of nucleus into the cytoplasm to participate in protein synthesis. So Gamow, G-A-M-O-W, discovered that a sequence of 3 nucleotides codes for one amino acid in the polypeptide gene. It means code words for amino acids in DNA and mRNA are nucleotide triplets, and each triplet form one codon. One molecule of DNA serves as a template for many mRNA molecules. Each of these serves as template for the formation of a large number of polypeptide genes. Okay. Then ribosomal RNA. Earlier we mentioned about three types: ribosomal RNA, tRNA, messenger RNA. We just learned about the mRNA. Now next we are going to read about ribosomal RNA or rRNA. Occurrence: the ribosomal RNA or rRNA constitute bulk for the cellular RNA, or 80% of the weight of total RNA of a cell. It occurs in ribosomes, which are nucleoprotein molecules. Types of rRNA. Inside the ribosome of eukaryotic cells, rRNA occurs in the form of particles of three different dimensions. These are designated as 28S, 18S, and 5.8S. The 28S and 5.8S molecules occur in large subunit, 60S subunit of ribosome, whereas 18S molecule is present in the small subunit, 50S subunit of ribosome. The 28S molecules of rRNAs have a molecular weight 1.5 to 1.8 into 10 raised to 6 Daltons, whereas 18S particles are only 0.7 into 10 raised to 6 Daltons. In prokaryotic cell, there are only 23S, 5S, and 16S rRNAs with a molecular weight of 1.2 into 10 raised to 6, 3.5 into 10 raised to 4, like that, 0.6 into 10 raised to 6 Daltons respectively. Inside the ribosomes, rRNA can remain folded. They are folded. Base composition of rRNA. rRNA differs in base content from tRNA and mRNA. It is relatively rich in guanine and cytosine. The base components in rRNA of E. coli have a molar ratio of adenine, uracil, guanine, and cytosine. Biogenesis of ribosomal RNA. Cells contain millions of ribosomes, and each ribosome contains several molecules of rRNA. The rRNA sequence encoding rRNA. The DNA sequence encoding rRNA are normally repeated hundreds of times, showing tandem arrangement. This DNA is called rDNA. It is clustered in one or more nuclei. Thus, this nuclear they just occur as tandem arrangements. Nucleolus and ribosome producing organ. The part of chromosome containing rDNA is called nucleolar organization. In bacteria, about 10 to 20 histones are associated with RNA synthesis, but in eukaryotes, 210 to 2100 tightly clustered rDNA histones are involved in RNA synthesis. The process of rRNA synthesis involves following steps: synthesis of rRNA precursor. rRNA precursor is synthesized on DNA histones as short fibrils with the help of RNA polymerase enzyme. Under electron microscope, the fiber appear as a chain of Christmas tree, which is about 100 or so fibrils show gradual increase in their length from one end of the Christmas tree trunk to the other. The shorter fibrils are located closer to the initiation point of transcription, and the longer fibrils are close to completion of transcription. The completed RNA fibrils are called rRNA precursor or primary transcripts. Spacer DNA, locus promoter region, 45S RNA chains. Christmas tree arrangement. And processing of rRNA precursor. Of the 4 rRNA molecules, 3 are synthesized in a single primer transcript, also called pre-rRNA. 5.8S rRNA is synthesized from separate RNA precursor outside the nucleolus. The primary transcript for rRNA is a 45S molecule of about 13 kilobases consisting of 1300 nucleotides. The processing of 45S rRNA occurs inside the nucleolus when it is tightly associated with the protein forming ribonucleoprotein purines particles. And rRNA undergoes 4 principal cleavage events as follows. First cleavage of primary transcript occurs at site 1 and removes 5' terminal leader sequence and produces 41S intermediate. Second cleavage occurs in 41S intermediate at site 2 or 3. Cleavage at 3 generates 32S intermediate. Then final processing steps involve separation of 32 intermediate into 28S and 5.8S sections of rRNA. The 28S and 5.8S with proteins form large ribosomal subunit. Processing of ribosomal RNAs, the eukaryotic transcription unit that include the genes for the three largest rRNA occurring in multiple copies arranged in a tandem arrays. Non-transcribed spaces separate the unit. Each transcription unit includes the genes for the three rRNAs and 4 transcription spaces. A single long primary transcript of large pre-rRNA in a sedimentation coefficient of about 45S. C and D DNA processing yields mature 18S, 5.8S and 28S RNA molecules. RNA cleavage actually occurs in series of steps and positions of cleavage is shown by some vertical arrows. Synthesis of and processing of 5S rRNA. 5S rRNA is about 120 nucleotides long. It has extra nucleolar origin. The rDNA for 5S rRNA is located outside the nucleolar organization. It also exhibits tandem arrangement of nucleotides or genes. The process of transcription of 5S rRNA requires RNA polymerase III enzyme and 3 factors, namely TF2A, TF3B and TF3C. TF3A is also there. So TF3A is specifically required for initiation of rRNA. The function of rRNA is not known, but the recent evidence suggests that one of the subunits of rRNA serves to release mRNA from DNA. Largest molecules of rRNA, 23S in prokaryotes and 28S in eukaryotes, is a riboenzyme that catalyzes synthesis of peptide bonds between the amino acids. It functions as peptidyl transferase. And transfer RNA, next one is transfer RNA. 5S pre-rRNA gene cluster, a pattern of tandem repetition, and in the middle space arrangement of sequences in the non-transcribed space region between the 5S genes. Okay, transfer RNA, tRNA. The transfer RNA is a family of about 60 small-sized ribonucleic acids which can recognize the codons on mRNA and exhibit high affinity for 21 activated amino acids, combine with them and carry them to the site of protein synthesis. tRNA molecules have been variously termed as soluble RNA or supernatant RNA or adaptive RNA. tRNA is about 10 to 15% of the weight of the RNA of the cell. Characteristics of tRNA. tRNA molecules of following characters. tRNA molecules are smallest, containing 75 to 93 nucleotides. Each tRNA molecule is a 3' OH terminus and a 5' monophosphate terminus, not 5' triphosphate as in outer case of other RNA molecules. Their sedimentation coefficient is 4S and the molecular weight is 24,000 to 31,000 daltons. This polynucleotide chain undergoes secondary and tertiary foldings because of internal complementary base pairing. As a result, the tRNA molecule acquires a precise L-shaped three-dimensional configuration resembling its polynucleotide chain undergoes secondary and tertiary foldings because of internal complementary base pairing. As a result, the tRNA molecule requires a precise L-shaped three-dimensional configuration resembling a twisted clover leaf. In 1965, R.W. Holle proposed a clover leaf model of tRNA molecule. For this, he got Nobel Prize with Khorana and Nirenberg in 1968. Some of the bases of two arms are foiled and coiled over one another. The 3' end of the polynucleotide chain of tRNA is called acceptor end. It ends in CCA base sequence. This represents site for the attachment of activated amino acid. The 5' end of the chain terminates with guanine base. The band in chain of each tRNA molecule contains a definite sequence of 3 nitrogenous bases which constitute the anticodon. It recognizes the codon on mRNA. Next, we are going to learn about clover leaf model of tRNA. Very famous model about the tRNA structure. According to the clover leaf model, 4 different regions or special sites can be recognized in the molecule of tRNA. These are amino acid arm or amino acid acceptor arm, AA arm or acceptor arm, It is double helical and stem-like. It possesses both the 5' and 3' ends of the molecule. It has 7 base pairs and the unpaired 3' terminal region always has a base triplet CCA with a OH at the end. The COOH group of a specific amino acid joins with the OH group of adenosine base of CCA in the presence of ATP, forming amino acyl tRNA. It is common to all tRNA molecules. Therefore, 3' end of acceptor arm is called a carrier end. An anticodon arm. It is a loop-like structure consisting of 7 to 11 unpaired nucleotides. It lies opposite to the AA arm, amino acid arm, anticodon arm. 3 of the 11 nitrogenous bases of this ribonucleotides are complementary to the one of the triplet codon of mRNA molecule. Since the base triplet on mRNA chain is called codon, its complementary base triplets on tRNA molecule is termed anticodon. Anticodon reads its appropriate codon on mRNA and temporarily binds to it. Therefore, the terminal end of this arm is called the recognition end. Then DHU loop, dihydrouridine loop. It is also a loop-like arm. It has enzyme recognition site and binds to a specific amino acid activating enzyme, which catalyzes the union of specific amino acids to tRNA molecule. Then T-Cy-C loop. It is also a loop-like arm of the tRNA. It is a site for attachment to a ribosome. This site is common to all the molecules of tRNA. It is found in 5 base pairs and has terminal loop with 7 nitrogenous base. Variable arm. It contains from 4 to 21 nucleotides. This portion. The T-Cy-C loop, which is thought to interact with ribosomal RNA, is at the corner of the L. The T-Cy-C loop, which is thought to interact with ribosomal RNA, is at the corner of the L. All bases except 5, 16, 17, 20, 47 and 76 are stacked, even though half of the bases are not in double-stranded helical regions. Even the bases of the single-stranded 3' terminal segments, except for the terminal adenine, are stacked. There are 9 base pairs that are not shown in this clover leaf model. They are called tertiary base pairs because they are responsible for the 3-dimensional folding. Only one of this is standard pair is a standard Watson-Crick pair, GC pair. In addition to the tertiary base pairs described, the tertiary structure is stabilized by hydrogen bonds between bases and different ribose units and between ribose units in separate nucleotides. So when we try to have a quick recap about all this, a cloverleaf model is a eukaryotic alanine tRNA molecule from yeast as determined by Holle. Then a modified nitrogenous base present in tRNA are 9 used in pseudouridine, DHU, DIHU5 dihydroxyuridine. The usual bases are adenine, uridine, cytosine, and guanine. tRNA structures. Each tRNA molecule possesses 4 special sites represented in the acceptor arm of tRNA represents site for the attachment of amino acids. D loop represents specific site for the recognition of amino acids. Anticodon loop bears anticodon recognition, anticodon, and T-Cy-C bears ribosome recognition site. Next extension, structural similarity in tRNA molecule. 2 observations make it like that all tRNA molecules are structurally smaller. Most of the tertiary base pairs are formed from bases that are reserved in all tRNA molecules. Different tRNA molecules can co-crystallize, forming uniform crystals. In a low solution X-ray crystallographic analysis of this mixed crystals does not indicate that 2 types of molecules are present and yield molecular dimensions that are the same as those of the single kind of molecule. This suggests that all tRNA molecules are structurally similar. Then unusual bases in tRNA. In addition to the usual bases of tRNA like cytosine, guanine, adenine, and uracil, each tRNA molecule has several unusual bases. Some of them are pseudouridine, eosinic acid, methyl guanine, methyl aminopurine, etc. The presence of these rare nucleotides, unusual nucleotides, does not affect the pairing of tRNA with mRNA. This presumably will probably prevent intramolecular base pairing in the opposite tRNA loop or help in the recognition of amino acyl tRNA synthase enzyme. tRNA molecules occur in both active and inactive forms. The inactive molecules of tRNA lack the CCA sequence of nitrogenous bases at 3' end of the chain, either in full or in part. By the addition of this nucleotide sequence, an inactive tRNA molecule is activated. This process is governed by cysteine triphosphate (CTP) and adenine triphosphate (ATP). Transcription and processing of tRNA. tRNA molecules are synthesized at particular regions of DNA. In the beginning, each tRNA new molecule exhibits complementarity in base composition and sequence to its particular cistron as the DNA. But in certain specific points, the nitrogenous bases are altered through enzyme action after the complete tRNA molecule has become transcribed from DNA. About 40 to 80 genes of cistones are involved in tRNA synthesis in bacteria and 55 in Drosophila. This appears to be scattered in chromosome in E. coli. The transfer RNA plays a key role in protein synthesis. It picks up a specific amino acid activated from the cytoplasm, carries it to the site of protein synthesis and attaches itself to ribosome in accordance with the sequence specificity by mRNA. Finally, it transmits its amino acid to the polypeptide chain which is being synthesized. Cells need at least 32 tRNA to recognize amino acid codons, but some cells use more than 32 tRNA molecules. Then extension ribozymes. American biologist Thomas Sich and his colleagues in 1983 discovered that certain pieces of RNA acted as enzymes or biocatalysts to cut selected pieces out of strands of viral RNA. These were named ribozymes, and he got Nobel Prize for this chemistry of this discovery in 1989. Like enzymes, ribozymes are not used up during reaction. Therefore, they can continue to snip up one virus after the next until all the viruses in the culture are destroyed. Functionally, there are two types of ribozymes. Ribozymes that carry out intramolecular and catalysis and catalyze reactions on themselves. They have folded structure. Ribozymes that carry out intermolecular catalysis and act on other molecules without themselves being changed. Other important cellular reactions are catalyzed by ribozymes. The maturation of hnRNA, heterogeneous nuclear RNA, the chipping out unnecessary sections from primary RNA, transcripts of ribosomal RNA synthesizing genes. 23S and 28S rRNA of ribosome catalyze the formation of peptide bonds during synthesis of polynucleotide chain in a prokaryotic and eukaryotic cell. Catalytic role of ribosomal RNA are established by Sich in early 1980s while studying tetrahymena ribosome and by Sidney Altman from the study of RNAsP. The final support for the catalytic role of RNA came in 1992 from Harry Noller and his colleagues. Scientists are working to use ribozymes therapeutically. Other types of rRNAs. In addition to these three types of RNAs, following types of RNAs are also found. Small nuclear RNAs, snRNA. These are small RNA molecules formed of 100 to 215 nucleotides. These are formed in the nucleus of eukaryotic cells. They are important for RNA replication and processing of long newly transcribed nitrogenous nuclear RNA molecule. They are genetic RNAs. In some viruses, RNA is the genetic material. It carries the hereditary blueprint. In some viruses, hereditary RNA is single-stranded, but in others, it is double-stranded. Enzymatic RNA or ribozymes. Thomas Sich and Sidney Altman independently discovered that certain RNAs act as biological catalysts. These are called ribozymes. These are associated with the maturation of hnRNAs, heterogeneous nuclear RNA. Even the 23S and 28S ribosomal RNAs of ribosomes are found to catalyze the formation of peptide bonds during synthesis of polypeptide chains in prokaryotic and eukaryotic cells. And functions of different types of RNAs. RNAs are essential for protein synthesis. mRNAs receive genetic information from RNA by transcription, comes out of the nucleus into the cytoplasm and translates the coded information into a protein at a determined sequence of amino acid. tRNA picks up amino acids from cytosol of cell and brings it to mRNA template at a specific position for polymerization. tRNA with proteins from structural component of ribosomes and provides surface for attachment for mRNA and tRNA. In TMV, HIV and influenza virus, RNA acts as genetic material. Small nuclear RNAs, snRNAs, are formed during DNA replication and are essential for DNA replication. Ribosomes act as biological catalysts. These are needed during maturation of heterogeneous nuclear RNAs into functional mRNA molecules. Some ribosomes control peptide formation during synthesis of polypeptide chain.